Good morning or good afternoon webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Advancing Personalized Oncology with Transcriptomic Profiling and Mechanistic Modeling. This webinar is sponsored by QIAGEN. Our speaker today is Dr. Moritz Schütte, Head of Next Generation Sequencing Analysis at Diagnostics in Berlin. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of your webinar presentation. We will ask our speaker your questions after his presentation has concluded. And also, if you look at the bottom tray of your window, you will find a series of widgets that can help enhance your webinar experience. So with that, let me turn it over to Moritz. Please go ahead.
Thank you, Julia, for the kind introduction. Good morning and good afternoon also from my side. First of all, thanks a lot to GenomeWeb and QIAGEN for the opportunity to speak today, and also many thanks for the support during the preparation and test run. So first, I would like to give a brief introduction of our company. So we are from Berlin, Germany. The company was founded as a spin-off of the Max Planck Institute for Molecular Genetics in 2011. And we're working mostly in the field of personalized medicine. So we do have a diagnostic test, a comprehensive molecular tumor analysis called CMTA, which I will present today. And we do have an NGS lab, of course, with strong expertise in bioinformatics. And we're also not only working on commercial projects, but we also participate in a number of national and international research projects like IMI or Horizon 2020. And last, what I will also show later is that we are developing a mathematical model, which is already working, which is called ModCel. And this is used to perform drug predictions for cancer cells. And the comprehensive molecular tumor analysis and the ModCel model are the main topics of today's talk. So why do you need tumor diagnostics? So different to many other diseases, cancer is a very heterogeneous disease. So not only heterogeneous between different affected organs, like breast cancer different from lung cancer, but even within a single cancer entity, like, for example, here shown colorectal cancer, there's a huge heterogeneity due to the underlying genomic alterations, which lead to a complex patient-specific pattern. Like in colorectal cancer, as you can see here, there's most recurrently mutated genes IAPC or TP53, but then you also have mutations in KRAS or NRAS, and many different combinations of these, making it a very complex and patient-specific disease. So you need a specific diagnostics to account for that heterogeneity, and this is not only due to genetic changes, but also due to epigenetic changes, which could be reflected or are affected in the transcriptome. So again, from colorectal cancer, here are two examples for transcriptomic patterns. On the left, you have different colon cancer subtypes, which can be made visible due to transcriptomic clustering, and those have associations to prognosis or could have associations to therapy. On the right, you see a gene expression signature for the response to cetuximab that was found in the same study. Cetuximab is a target of EGFR, and the known biomarker is mutations in RAS or KRAS, NRAS. And here we could show that a gene expression signature could even outperform mutations in RAS or KRAS and NRAS in terms of accuracy for predicting response to cetuximab. So taking the transcriptome gives even a more complex pattern and more information on the heterogeneity and on the disease itself. And that heterogeneity within cancer is also reflected in the response rates to drugs. Every patient is different, every tumor is different, if you look at response rates of very known biomarkers, here on the left, you see that the median response is only about every second patient responds. And the duration is, can be varying quite a lot from some markers and the treatments leading to a long-lasting response, while others are in the range of months to only a few years. And so you really need a good diagnostic to first identify the main changes before choosing best treatment. Now, saying that there are changes on the DNA, on the genetic level, and also on the transcriptomic epigenetic level, you can summarize that you would need both to properly characterize your cancer. So on the left, you have the genomic changes, and on the right, that's the functional changes from RNA, which could give you additional informations like isoform, EGFR, variant 3, or RTE, which are oncogenic isoforms, gene fusions. And then you get also the abundance by gene expression measurements of all the genes, which you can do on the tumor or the tumor and the tumor environment, like the T cell. And in our diagnostic, we use DNA combined with RNA. So as I said, the tumor microenvironment is becoming increasingly relevant with immunotherapies. Very known are checkpoints like PD-L1, which can be targeted, but there are a number of other checkpoints like CTLA4, TIGIT, LAG3, and those you can determine as targets via RNA-seq by looking at the bulk tumor, which consists of tumor cells and surrounding and infiltrating immune and other cells. Speaking of CTLA4, when you, as an example, there's an increasing number of clinical trials going in the direction of having combination therapy, not only a single drug therapy, so one biomarker, one drug, but for example here for CTLA4, ipilimumab, you have a number of trials looking at treatments in combination. And other examples is, for example, BRAF plus MEK inhibitor treatment in melanoma, but there are also other examples. And so going to more complex treatments would also need a more comprehensive diagnostics first. For this clinical need, we do have two solutions addressing those. So as I said, one is our comprehensive molecular tumor analysis based on whole exome plus transcriptome sequencing. So while others do some sort of small diagnostics, single markers or a panel, we try to get the most advanced diagnostics and using transcriptome sequencing. And the second, which I will show later, is still under research and development, is a mathematical modeling, which is actually a complementary system to the diagnostic. So first, what we do on our diagnostic, that's similar to many other diagnostics, so there's clinical consultation, then we prepare nucleic acids, DNA and RNA, and libraries from either FFP or frozen biopsies. We do have our own in-house bioinformatics pipeline, and at the end, there's a curated report summarizing the findings plus potential treatment options, which could go to the molecular tumor board or an oncologist. And the report summarizes all those key features, mutations, copy number changes, for example, from DNA, but then also rearrangements and gene expression profiling on genes or gene signatures from RNA-seq. Well, technically, this approach could be done on any tumor, so still the main focus at the moment are those tumors which already have received the standard of care and are now in need of a new treatment option. As I said, we use RNA-seq a lot, so there's one part where we do actually detection of variants like gene fusions and gene expression from RNA-seq, but we also integrate always DNA and RNA, use it as an orthogonal validation and for functional predictions. So on the bottom left, you have one example of a missense variant in the gene BOB1, which is a relevant tumor suppressor in uveal melanoma or meningioma. And this is a missense variant that hasn't been described before and might have been classified as a variant of unknown significance. But when you look at the RNA in the same sample, you see that the missense variant obviously leads to a novel splice site, and you see a novel splice junction from exon 4 to exon 5, which in fact then leads to a frame shift. And such, it's clearly a pathogenic mutation and not a variant of unknown significance. The second example, on the right would be to integrate copy number detection together with gene expression and overexpression analysis. So that is an example where we determine an amplification, which a locus which contains the estrogen receptor. And then at the bottom, you see that the gene itself, compared to a cohort of tumors of the same tumor type, is really highly upregulated. So this is a positive example where the expression validates and supports the finding of the amplification, but we also use it in the opposite way, that you sometimes have a locus with more genes, and the expression can tell you which of them are actually truly upregulated and which maybe aren't, still amplified but not upregulated, and thus probably not the most intuitive target for the treatment. Last part is to use bulk RNA-seq. As I've said before, from bulk tumor, you have tumor cells, but you also always get some surrounding cells, some infiltrating cells. And characterization of tumor microenvironment actually has impact on some targets, so angiogenesis, for example, or immune checkpoints and hypoxia. So it's necessary now to characterize the tumor microenvironment to look for, better characterize the tumor and to see for those treatments. For that, we do have a couple of different approaches. One would be to deconvolve the tumor and try to quantify really if the proportion of different cell types within the sample that we get. That's a deconvolution. Then we use cell type specific signatures. So there are signatures for T, B cells, neutrophils, and so on. And those will be analyzed across a cohort of tumors to see where the sample would position within that cohort. And last would be to go for single individual markers like CD68, which is a macrophage marker. And from RNA-seq, you can basically quantify the expression of any gene of interest. Then, having analyzed the sample and all the relevant alterations, the next step would be to link them to actual treatment. And for that, we have a dual approach. So one is to have in-house a database of drugs and drug targets. And the second part is that we use QIAGEN QCI, Clinical Insights, to get annotations for the alterations that we find. So when you use QCI, the standard way would be to upload a VCF file. The VCF file can not only contain variants, but there's also the possibility to include gene rearrangements, copy number changes, and other alterations. You can do that using an API. That's the typical result that you get once the sample is uploaded. So you have an evidence level sorting. At top, you have a really tier 1 finding, ETVF runs 1 fusion, and then this is mapped to the actual disease, in this case acute lymphoblastic leukemia, and you get some functional annotation like gain or loss for those variants. Now, if you go one level deeper and looking at one individual finding, again ETV6 rungs 1, you get a plethora of additional information. So there are guidelines for approved drugs like NCCN or EMA. You get literature evidence on the functional predictions and preclinical studies. There's a section on clinical trials, which you can filter for using the country you're interested or the location. And then what I also kind of like, which is a fairly novel feature, is an expert annotation from the company N of 1, which was then acquired by QIAGEN. So they perform manually curated annotations for specific variants. And this is kind of nice if you have a variant which is a bit rare, so you don't have to do any literature search, but get a pre-annotation with the domains with some publications on those variants. And so we use this system as part of our pipeline to get clinical information and therapy information into our reports. So as a first real-world example, we are not only doing this using exome and transcriptome, but also we're doing diagnostics for using transcriptome only for a certain entity, which is acute lymphoblastic leukemias, pediatric acute lymphoblastic leukemias in relapse. And the idea is here to get a stratification based on high-risk group, which are characterized by p53 mutations or KMT2A rearrangements, and then also scan for other alterations which might lead to some targeted therapies if needed. So the setup for this diagnostic is to do paired end 100 RNA-seq with at least 100 million reads. And from that, we perform mutation analysis, gene fusion detection, and gene expression profiling. So to validate variant calling, we have used samples where patients where we did have DNA and RNA sequencing, so similar to that CMTA approach before. Of course, with RNA, you can only detect the variants which are actually expressed. But looking at a number of samples, you will see that any clinically relevant mutation is in fact expressed. So we focused on those and performed a scan on a number of different entities. And I actually found that it's a very reliable and accurate method, depending on the coverage. That's the main important parameter. But if you look at the top right, so those are very relevant genes for acute lymphoblastic leukemia, like RAS, FLT3, ETB6. All those genes are fairly well expressed. So the red line would correspond to 5 FPKM, which is idealized with 100 million reads, something like 50x coverage. And to summarize that project with some actual data, on the left you see the pattern of alterations we have of around 80 samples. So some mutations in green, missense mutations, and we have a lot of gene rearrangements, some fusions in this purple. And on the right, you can see a comparison to some published data for relapsed ALLs. So on the top right level, you have the reference cohort in blue and our cohort in red. And in the panel below, our data is split into mutations and gene fusions. And you see that for the most abundant genes, it's fairly similar for KRAS, NRAS, CREB-B, or p53. The most relevant genes, we find them in similar quantities. So as a good proof of principle, that is a really nice diagnostic, which is now applied for pediatric leukemias. So coming to the second part is the mathematical modeling. So modeling is actually a concept which is used in many other areas of life, like weather forecast, or if you do some industry design, car design. To reduce risk and make mistake or avoid mistakes, modeling is often done first, but not yet in this manner in health. And so the idea is here to model a patient before the drug is given to identify the best drug for the patient. So the way the model is designed, it's an ordinary differential equation model of the main signaling pathways in a cancer cell. So currently it's 46 signaling pathways, around 800 genes. And then we can model any drug with some molecular mechanism, so any targeted drug where we know where it binds. So it cannot be done for chemotherapies, but for any targeted small molecules or antibodies. The data that it needs is some gene expression data or genomic alterations. So in fact, it's a complementary system to the CMTA. So do a diagnostic first, use that data, and model the patient and get a report on drug sensitivity. To give an example here, how that would look like. So there's a generic model on the left, which is just a wild type cell. And on the right, you would initialize that with patient-specific data. So in this case, you would add a mutation, KRAS G12V, and you would put it in such an abundance corresponding to the variant allele frequency in the RNA-seq, so that you have the wild type and the mutant. And then this is modeled, and it will be modeled just as a cancer cell, and then as a cancer cell plus some drugs and some drug concentrations, so that you would, at the end, get an in silico response curve, which is shown on the right, together with an IC50 value to identify drugs for which it might be resistant in this particular patient-specific cancer cell, or it might be a responder. Since it's a computational system, it's of course possible to not only perform single drug experiments, but also to use any combination of interest. And we could show that using combination of two drugs, this mathematical modeling, the mechanistic model is with a correlation that you see on the right. It's within the same ballpark as some reference statistical models like Bliss independence and high single antigens. So the predictions of that model are similar at the moment to the best reference models. The application areas, so as I said, it could be used for individual patients to perform a drug screening and then give those values to a tumor board or an oncologist. Other areas would be virtual clinical trials, so that you actually take a cohort of patients, perform drug screening, and try to identify responders and groups so that you can find biomarkers of response. This could be done for novel drugs. Another option would be to use that again on already existing drugs to reposition them to a different cancer entity or to find a better group of patients who might respond. And the last part would be similar to, for example, a knockout experiment, so that within the signaling network you could knock out certain genes and perform a drug target identification in a preclinical setting. Now I'd like to introduce one pilot translational study where both approaches have been used. It's called Treat 20+ due to a prior project, but in fact it was around 100 metastatic melanomas. So those are melanomas which had received standard of care before, which could be BRAF treatment or immunotherapy. And then they were recruited into that study. So they came from different subtypes of melanoma. There's cutaneous melanoma on the skin, the acral and mucosal melanoma. And here in this project, we also had a quite large cohort of around 40 patients which had uveal melanoma, which is a rare melanoma affecting the eye. And for those patients, we performed bulk RNA-seq and whole exome sequencing together with the Max Planck Institute for Molecular Genetics and the Charité. And then those patients were analyzed and modeled, and the analysis results went to the Charité Molecular Tumor Board, where we discussed them with them. So that was, for us as a company, really helpful and great to have this conversation and interaction, and to see how patients are discussed at the tumor board and how oncologists read and discuss the data. And for us, it was a great opportunity to improve the way reports are written and how we present data. And then patients got treated, and there's one publication already out, and we are currently preparing some more publications from that project. So that's a typical example how the data was transferred to the tumor board. So as we learned, there's only very limited time, so it had to be very compact. There was one page with key findings. This is an example of a uveal melanoma, so with mutations in GNA11 and SF3B1. There are some upregulated genes, MDM2, there's a digital karyotype, and then some immunotherapy relevant findings summarized on a single page. And then the report had numerous, like 20 pages with backup and more detailed data for the project and the interested physician. As an add-on to those clear findings, we always performed microenvironment profiling, as I have shown before, comparing a sample to a cohort of samples shown on the left for the main relevant cell types that are there from publications and other sources, for neutrophils, for macrophages, for T, B cells, Tregs, and so on. And then we looked at all the different ICI markers, PD-L1, X3, TIGIT, and others, to get the best diagnostic for the patient and most information. If you look at the entire project, there's numerous different applied biomarkers that were detected and discussed. So there are simple mutations like GNAC, GNA11, NRAS, different BRAF mutations, Notch or KIT. Then there are a couple of copy number changes which became interesting, like NF1 or P10 loss. We had patients with CDKN2A loss or ESR1 amplification or YAP amplification in the Hippo pathway. And then there are also a couple of markers from RNA-seq, like PD-L1 or IDO1 expression. We had a couple of gene fusions of interest, MET overexpression. And we also had a larger cohort of patients which had ALK-RTE, so alternative transcription initiation in codon before exon 20, which had been shown to be an oncogenic isoform in very few cancer types, and melanoma is one of them. And so we had a couple of patients who actually went into crizotinib treatment based on ALK-RTE. To give you two examples, that's case example 1, which is a patient which was wild type in RAF and BRAF and NRAS, and had received some pre-treatment with different immunotherapies, and then became, was recruited for the project, and we identified an NTRK fusion here. So the patient went into a clinical trial and received treatment with LOXO 101. And then the patient responded for approximately a year, which is quite a long time for metastatic melanoma. Second case example, it's skin melanoma, the patient which had a very rare mutation in BRAF. So this typical mutation that's well known is codon 600, so V600E, for example. And here it was an insertion in codon 599, for which there was only little evidence. So there were a couple of case studies from other tumor types. And based on that, the tumor board decided to try a BRAF inhibitor on the patient. And in fact, the patient responded for around a year again. And after that, there was a new biopsy taken and analyzed that you can see on the bottom left. And in that biopsy, that's a sashimi plot, so-called, from the exon junctions in BRAF. And you see that in the most recent biopsy, which is the red one on top, there's a skipping junctions. So a junction from exon 1 to 9, skipping exons 2 to 8. And this had been observed before for resistance to BRAF inhibitor treatment for BRAF V600E mutations. So showing that the treatment actually worked and that the stress and had pushed it in a way that this isoform has evolved as a marker of resistance. Now, in addition to those genetic alterations, there was modeling for the patients, and this is a typical modeling report. So in the top center, you would see a drug response curve that is done then for different drugs, which are screened across for the patient. You have some effective drugs listed, and then you would also get from different readout components of the model pathway activation for the patient to get a better information on the pathways which are activated due to the underlying mechanism. As one last example for modeling in conjunction with genetic alterations, it's a patient with BRAF V600E mutation and, for example, MET overexpression shown on the left in the pathway visualization. When the patient was analyzed using the modeling, it was observed that there's quite a small EC50 value for vemurafenib, so based on the BRAF mutation, which makes sense. But there's actually a resistance against MET, which can also be explained by the underlying BRAF mutation, which is downstream of the MET kinase. So this makes sense in a sense of a resistance and a response curve from the same patient. And with that, I would like to summarize that I've shown you those two approaches. So one is a comprehensive molecular tumor analysis, a diagnostic using whole exome sequencing and transcriptome sequencing, and with the advantage of identifying additional targets using transcriptome and also to quantify the tumor microenvironment. It's already used by some tumor boards. And the second part was about the mechanistic modeling, which is able to mimic a cell, an individual cell, and to predict responses to single drugs and drug combinations. And maybe one last word as a future is a digital twin concept, so that every patient would get his or her digital twin as a model of itself to simulate any drugs or any treatment decision, not only for cancer, but as a future also for other drugs, and has this as part of the health care. But that's sort of future. With that, I would like to thank everybody for supporting me. Thanks for listening, and to GenomeWeb and QIAGEN for all the help. Thank you, Julia.
Yeah, thank you very much.
Be happy to take some questions.
Thanks. Yeah, thanks very much, Moritz, for this very nice presentation. We do have a lot of time for questions, so just as a reminder, if you have a question, please type it into the Q&A panel. Also, we would like to ask our participants to take a brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let's see what we have for questions here. There were a few technical questions regarding your tests that you mentioned in the first part of the talk. One was there a targeted panel for gene fusions, or were the gene fusions just derived from the exome panel?
Well, we do have validated RNA-seq for a number of gene fusions, but in principle, it's an open approach that could detect any novel gene fusion, which of course then has to be manually checked if it's, what the functional prediction or functional effect would be, but it's open to any gene fusion.
Okay, great. Someone would like to know if you have a comment on RNA extraction from FFPE samples. I'm not sure if you actually did that. How can any stability issues be overcome related to that?
Yeah, we've done that for quite some time. We are now using RNA exome as a kit for that. I guess there's no clear, easy answer. So we are cutting section ourselves and do the extraction. And I guess it depends very much on the quality of the RNA block. So that varies quite a lot to our understanding. So some FFP samples are pretty okay for RNA-seq, but we also have seen some which are sort of poor.
Right, and that's a related question about the ALL case example that you used, that you showed. Did you extract RNA from blood for that, and were there any stability issues?
We did not extract ourselves, so for that project, RNA was taken from the samples from the blast at the clinic, and we received the RNA directly.
Okay, great. Yeah, another technical question. Can you describe your approach for normalizing RNA-seq expression across samples that are sequenced over a period of time?
Across samples over a period of time. So,
yes.
I mean, what we do
normalizing,
is a normalization based on the median values. So we median center them like a TMM's normalization. And that is done across the entire cohort simultaneously, if that is answering the question.
Okay, yeah. I hope it does. Let me see what else we have. Some general comment or question from somebody. Since you're analyzing the exome, would you miss anything because you're not sequencing the entire genome?
Well, I guess most of the known biomarkers are in the exome, so surely there might be some structural variants which are hard to detect. We're doing transcriptome in addition, which somehow reflects, of course, epigenetic changes. And if you have something like enhancer hijacking, you would not likely detect by exome sequencing, but still you would see the effect based on huge overexpression, for example, in the transcriptome.
Okay. Another question relating to gene fusions. How reliably can you detect gene fusions if you use paired end 100 RNA-seq? Is that actually what you're doing?
That is what we do. I mean, we have validated that on some reference samples, so there it is reliable. Of course, there are numerous possibilities for RNA fusions, and of course, it's expression dependent. But for the main drivers, I would think it's very specific and reliable detection method. Okay.
Can you talk a little bit about ModCel? A few questions related to that. So one is do you use ModCel only for oncology, or could it be also used for other fields like rare diseases or other fields?
Well, the main focus at the moment is oncology. It's a model of a cell. So if there's any other molecular interaction, like a target molecule interaction, it could also be applied there. We are currently also participant in a project where it is applied on arthritis, on rheumatic arthritis, and we had a different project before on bone disease. So, I mean, there are some approaches to generalize that and apply it on different rare diseases where you have any molecular interaction, but the main focus, let's say, is oncology.
Oncology main focus, but arthritis and bone disease, you have tried it on that. Okay, great. For the in silico modeling, what data sets did you use? And is the data set of a patient's response and overall survival from clinical trials?
Okay, so the data that is used is, it could be at least, it should be some gene expression data. We can also add like proteomics data and other molecular data, which can be mapped to a function in the cell. So all that could be entered. It could be some loss of genes, which could be entered as not using that gene in the modeling. Second part was on clinical trials. So I've shown at the end that we had the clinical study on metastatic melanoma. So that data we currently summarize. Mostly it's preclinical. So it was part of a number of research projects. This project, Oncology, I've shown them in the beginning. So there We also had cell line and xenograph data. At the moment, we are part in one project called Campus Pro, where ModCel is applied and improved using transgenic models of lung and breast cancer. So to have a very clean model system and then improve it.
All right. Question related to the ALL study you presented. Would that also look at long non-coding or small RNA, or only other types of RNA?
Small RNAs we don't do. Long coding could be done in principle. So we're using ribo0 RNA-seq, so it also captures non-coding RNAs. If they are biomarkers, we can use them, but at the moment we have a sort of fixed set of known biomarkers for ALL that we look at specifically, but it could be extended also to long non-codings. Got it,
yeah. And question about the melanoma study, what was the turnaround time from submission of samples to actionable reporting?
Well, the turnaround that we as a company have is like 3 to 4 weeks. During the melanoma project, it was something between 2 and 6 weeks. I mean, for financial reasons, we also had to sometimes collect samples for a bit of a longer time to have them simply sequenced together. I mean, of course, with infinite financial resources, it could be something like 2 weeks, but the normal turnaround is somewhere around 3-4 weeks.
Okay. Here's a question. I'm not sure if you can answer that, but it's about that one specific patient example you showed, and that was what is the reason why the patient with a BRAF insertion T59TT received a BRAF inhibitor instead of a MEK inhibitor? I don't know if you can answer that. So.
I mean, that was not my decision, but it's a decision of the tumor board. If I remember correctly, there were some case studies where that was applied, and maybe also some preclinical studies, but I cannot give a definite answer. I think it was also that functional studies have shown that the insertion should act similar to BRAF V600E. So that is, I guess, the main reason.
Okay. And in how far do you use epigenetic data or epigenetic signatures for your clinical interpretation? Can you just comment on that, how much that's included?
It is included on not on a regular basis, but on a tumor type specific basis, and sometimes based on request by physicians. So we are also open to add some research-only part with certain signatures which might come up or for specific tumor types, like, I don't know, triple-negative breast cancer subtyping or similar.
Right. Coming back again to the fusions, how do you evaluate the function of an unknown fusion when you detect one?
I mean, options are, we do RNA-seq, so you can see actually the expression. So if there's a huge overexpression of the domain, or a similar fusion has been described before, but now with a different partner, that would be options to how to describe them.
Okay.
To look at the protein domains involved and if there are similar fusions around.
Yeah, and I think here's a related question, more wider, not just about fusions. Is it possible to detect other new molecular interactions using your approach?
Can you repeat new molecular interactions?
Yes.
I guess not. Maybe the modeling could do that if you would perform something like a knockout screen on those. That would be an option. From the diagnostic not, but the modeling could eventually give hints on some molecular interactions.
Provide some hints, yeah. And then can you maybe explain in a little more detail how it is decided which molecular targets, drugs to use changed? That's what the model does, right?
Sorry, could you repeat? There was a bit of noise.
How do you decide which molecular target drugs to use in response to genetic changes? Like, how does this modeling work?
Well, the drug target interactions are implemented in the model based on available knowledge on the drugs.
Right.
And a modeling of a patient could incorporate any drug that has been implemented in the model. So then you could perform a drug on any screen on any drug combinations that you like. It's just a matter of computational time and interpretation at the end.
Mm-hmm. Okay. There's one question about your company's business model, I guess. That is, how do you plan to monetize? Is it expensive to run these screens, and how do you plan to get them widely adopted in Germany and also elsewhere?
So for the diagnostic test, we hope that we can make some negotiations with, individual healthcare providers to have that on single entities first and get some reimbursement by that. For the modeling, I guess it's still a longer way, which would be more like a cooperation or on a payment through pharmaceutical companies for virtual clinical trials or similar. So that would be the business models for that, but it's still a bit on the research. Yeah.
Okay, let me see what other questions we have here. Can you talk a little bit about the QIAGEN software that you use? What was the value of using that as part of your testing service? Was it easy to implement the QCI into your existing pipeline, for example?
Yeah, so the setup was quite straightforward. I mean, they would require a specific VCF format, so there we had a bit of implementation. Otherwise, it's quite straightforward to implement it into a pipeline. The value of it for us, I mean, we're not a huge company, and is to have a weekly updated database of drugs, of clinical trials, and to outsource that part of keeping track of all the information, which is quite a lot for us, and also to get more functional information.
There was actually just a question about that. How frequently is the drug database updated? And so you just said that happens weekly?
I guess it's per week, but I'm not 100% sure. I think this is what I learned.
Okay. And what tumor types are you mostly analyzing these days? You gave a few examples. Are there others? Or?
No, I mean, it could be any. So it's not specific to any tumor types. I guess the ones that it's a mix of different ones. I mean, ALL, I've shown that we do this diagnostic as a really structured cooperation with the Charité, but otherwise it's random tumor types. I guess the ones which are most hard to treat and most likely relapse. Okay.
And what is the turnaround time just for the whole exome and transcriptomics analysis? Just that part?
So that's around, including the lab experiments, it's something like 3 to 4 weeks from sample to report.
Yeah, we had that question earlier, the total turnaround time. Yeah. Okay. Do you have any recommendations for anyone else who has a lab that's interested in integrating the QIAGEN knowledge base into their own pipeline?
I mean, you can get a trial account. That's what we did, and then upload a couple of samples and see how it works and what's the value of it. But I mean, we are kind of happy with it. So I don't have any personal recommendation how to do that, but I could recommend to use it. For us, it's very helpful.
Have you ever seen conflicting results between your in silico findings and the QCI ranking?
In silico findings and QCI ranking, Okay, I guess that's something that is really difficult to compare a ranking based on evidence levels. And I mean, we never really done that. Of course, we have compared in silico predictions with drug responses that we get from preclinical or clinical data. And there we sometimes see discrepancies, of course. But I guess it's not very helpful to compare in silico predictions using actual data versus some sort of annotation of findings. Yeah.
Right. Yeah, and lastly, what are some of the challenges you've experienced in implementing a precision medicine program like that in-house?
I think if you do that, there's of course a lot of ontology challenges, like how do you call the tumor types, how do you keep track of them in a way that it can later be used for QCI. So that's something that you have to keep track of. And then, of course, at the end, to evaluate the results is something that you have to take care of. I mean, not to copy them one by one, but maybe to still have some sort of system to look at them and to see that in the whole context of a patient. I think this is also something that is a challenge that you have to do, because you get all those different alterations, but then you get alteration-specific recommendations, so you have to combine them at the end again to get a final summary of the patient.
Okay. All right. It looks like this is all the questions we have for today. So let me thank our speaker, Dr. Moritz Schütte, and our sponsor, QIAGEN. And as a quick reminder, if you please look out for the survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, a link to an archived version will be sent to all attendees. So with that, thank you very much for attending this genome webinar.
Thank you from my side for attending. Thank you, Julia and